En Fundación UNAM seguimos trabajando por la cultura, la ciencia y la educación. Sin salir de casa, reflexionarás con los martes de ciencia a través de las conferencias Universidades por la Ciencia. Emitimos en conjunto con el Colegio Nacional y para disfrutar regresamos con la segunda edición de los ciclos de documentales de Jueves de Ciencia con la transmisión especial de TV UNAM. Además, conversamos sobre nuestra nueva realidad y sus afectaciones en distintas disciplinas a través del Corredor Cultural de Autonomía. Sigue nuestras redes sociales y no te pierdas ningún detalle en www.fundacionunam.org.mx.
Los alcances de la ciencia son cada vez más audaces e innovadores. Las nanociencias nos conducen al microcosmos, un mundo imperceptible para el ser humano, en donde se estudian, se diseñan y se aplican nuevos materiales y diminutas herramientas para usos prometedores. El avance de la nanotecnología brinda posibilidades de mejorar la medicina y la salud, así como de aportar opciones para el rescate y conservación del medio ambiente.
En Fundación UNAM seguimos trabajando por la cultura, la ciencia y la educación. Sin salir de casa, reflexionarás con los martes de ciencia a través de las conferencias Universidades por la Ciencia, que emitimos en conjunto con el Colegio Nacional. Y para disfrutar, regresamos con la segunda edición de los ciclos de documentales de Jueves de Ciencia, con la transmisión especial de TV UNAM. Además, conversamos sobre nuestra nueva realidad y sus afectaciones en distintas disciplinas a través del Corredor Cultural de Autonomía. Sigue nuestras redes sociales y no te pierdas ningún detalle en www.fundacionunam.org.mx.
Los alcances de la ciencia son cada vez más audaces e innovadores. Las nanociencias nos conducen al microcosmos, un mundo imperceptible para el ser humano, en donde se estudian, se diseñan y se aplican nuevos materiales y diminutas herramientas para usos prometedores. El avance de la nanotecnología brinda posibilidades de mejorar la medicina y la salud, así como de aportar opciones para el rescate y conservación del medio ambiente.
Bienvenidos a el ciclo de conferencias de Fundación UNAM y el Consorcio de Universidades por la Ciencia. El día de hoy tenemos una conferencia muy especial a cargo del profesor Philippe Meade de la Universidad de Oxford. Welcome to the Universities for Science seminar cycle organized by the UNAM Foundation. Today we have a very special conference by Professor Philippe Meade, who is in the University of Oxford and is a fellow of the Royal Society. The conference is on the modeling of a collective cell movement in biology and medicine. And it's a privilege and an honor to have you here. Welcome, Professor Meade, and we look forward for your conference. Voy a pasar la palabra a nuestro presidente de Fundación UNAM, quien es el que organiza estos ciclos de conferencias. I will introduce Professor Dionisio Meade, who is the president of the UNAM Foundation, who will give us a welcome remarks. Thanks very much to all of you, and we are glad to have you all here. Thanks.
Muchas gracias y bienvenidos todos. Es un gusto que nos acompañe nuevamente en esta reunión semanal de Consorcios por la Ciencia, y justamente la presencia hoy del doctor Philip Meade de la Universidad de Oxford acredita el acierto de haber diseñado junto con el Colegio Nacional, junto con la Universidad Nacional, con el impulso de Araxi y de Jaime, esta idea en donde nos vinculamos, pues ahora sí que superando fronteras, superando barreras de lenguaje, en este esfuerzo de divulgación del conocimiento y compartir, sobre todo, investigaciones en curso. Quiero empezar estas palabras felicitando al doctor Jaime Urrutia, que ayer fue designado pues, profesor emérito de nuestra Universidad Nacional, y que es un orgullo que desde su pertenencia en la Junta de Gobierno de la UNAM, en la Junta de Gobierno del Colegio Nacional, y que nos honre también como integrante del Consejo de la propia Fundación UNAM. Es para nosotros un orgullo que esté con nosotros, y nos felicitamos de que con su impulso podamos realizar pláticas como la que hoy se va a celebrar. Quiero nada más subrayar que la pandemia, que trajo consigo muchas dificultades, mucho dolor, mucha frustración, sí trajo también consigo oportunidades como esta, y que al compartir estas investigaciones en curso y al impulsarnos a conocer mejor nuestro planeta, el funcionamiento de nuestro organismo, el conocimiento de cada vez más y más sistemas como el que hoy se nos presentará respecto a los movimientos celulares en biología y medicina, acreditan que la ciencia va avanzando, que hay que hacerle caso a la ciencia, y que es una formidable oportunidad contar hoy con la presencia de un especialista de la talla de Philip, y de la presentación y moderación que nos hará hacer el doctor Kirill Cherednichenko, que ya nos ha acompañado en otras reuniones. Bienvenidos todos, y le damos la palabra al doctor Philip Meade. Gracias de nuevo.
Thank you very much, Professor Meade. Hello, everyone. I'm Kirill Cherednichenko from the University of Bath, and it's a special pleasure to introduce our speaker today, Professor Philip Meade from the University of Oxford. Professor Meade is a truly Oxford man. He obtained his undergraduate degree from Balliol College, Oxford in 1982 and DPhil in 85 under the supervision of Jim Murray on morphogenetic pattern formation. He was briefly a mathematics tutor at Eton College, following which he held a junior research fellowship at Wolfson College from 87 to 88, and an assistant professorship at the University of Utah in 88 to 90. He then moved back to Oxford for a lectureship in mathematical biology and the tutorial fellowship at Brasenose College, followed by professorship in 98. In 2005, he was appointed to statutory professorship in mathematical biology with the professorial fellowship at St. John's College, Oxford. This happened just as I myself left St. John's after a 4-year junior research fellowship there, although I'm not completely certain there is a connection between the two events. I recall many pleasant encounters with Philip during those wonderful 4 years. During that time, since in 1998, Philip started leading the Wolfson Center for Mathematical Biology at the Mathematical Institute in Oxford. Philip's research includes the mathematical analysis of tumors, wound healing, and embryonic pattern formation, and the theoretical analysis of these models. Throughout his career, he has supervised 62 PhD students to their doctorate and is currently supervising 12 students. In 2015, he was elected a Fellow of the Royal Society, and in 2010, he gave an invited talk at the International Congress of Mathematicians, speaking on mathematical aspects of tumor technology. Philip has been on editorial boards of a large number of journals, including editorship-in-chief of the Journal of Mathematical Biology, and he has been elected member of a number of prestigious learned societies, including notably la Academia Mexicana de Ciencias as a miembro correspondiente. I'm sure Philip will share with us today some insights into his approach to mathematics and interdisciplinary research. So I guess I'll be stopping here and passing it over to Philip for his lecture on modeling collective cell movement in biology and medicine. Just one more note before he starts. Questions are very welcome in the chat in any of a variety of languages: Spanish, English, French, Russian, or Ukrainian. Over to you, Philip.
Thank you, Kirill, for that very kind introduction. And thank you for the invitation to give this talk. So I just get this set up. Whoops. Okay, so I don't know why it's not working. I think. So, okay, so can you?
It's not full screen yet, Philip.
It's not full. Yeah, is it okay now?
Here it is. Now it's okay.
It's okay now. Yeah. So sorry about that. And I'll just get one of these things here. Okay. Yeah. So thank you, Kirill, for that very kind introduction, and thank you to UNAM for this very kind invitation. And so I'm going to be talking about modeling collective cell movement in biology and medicine. And I would like to dedicate this talk to Professor Edmund Krampen, who was a former graduate student of mine and who passed away very tragically and unexpectedly 3 days ago. Edmund was Rowden White Chair of Systems Biology at the University of Melbourne, and he was an outstanding academic and an incredibly nice, friendly, helpful individual, and it's a tragic loss of his very early passing. So I will dedicate the lecture to him, and in fact some of the work I'll talk about is based on work that Edmund pioneered as a graduate student. So I'm going to talk about collective cell migration, and this is a very common thing. It happens in normal development in the embryo. It happens in repair when we need to, we get a cut and there's a hole and the cells need to move in. And it happens in disease like cancer. So my talk, I'm going to look at two examples of collective cell movement. One is going to be in developmental biology, and one is going to be the formation of new blood vessels, which happens in wound healing and in tumor growth. And the first one is going to be very interdisciplinary, and the second one is going to be more mathematical. So cranial neural crest cell migration. So first of all, I should point out, this is interdisciplinary work with experimental colleagues at the Stowers Institute for Medical Research and my collaborator Ruth Baker and a number of exceptional graduate students, Louise Dyson, Linus Schumacher, and Raza Ginunate, all here. So neural crest is the name given to structures that start off near the backbone and cells move out from there to various areas in the body, like, for example, the heart and pigment cells. And there are 4 different neural crests. And there's been a huge amount of mathematical modeling done in this area. This is just some of the people who've worked in this area. So we're going to be looking at cranial neural crests. So this here is the head of an embryo. This is a chicken embryo. And this is the backbone, the spine. And we are looking at cells that come out of here, R4, and they migrate to something called BA2. And they form the lower part of the jaw of the body. So here, more schematic, here is the, from head to tail, the head down to tail, and the cells are moving out like this. And so if you look at cross-section, they're moving like this. And importantly for studying this is that, well, there are many birth defects that arise due to problems in the neural crest. And the neural crest, cranial neural crest, is very similar to some cancers. So it's important from a fundamental biological point of view. It's also important from a medical point of view. So our biology colleagues can track the cells and they can measure as the domain grows. So the domain starts off at, roughly speaking, 300 microns, and over about 24 hours, it grows to about 1100 microns. And you can see here its curve, and it is 3-dimensional, but we do an abstraction. So we assume that the domain is flat and it's a rectangle. So basically, the question is the following. If you have a rectangular domain and cells are coming in at one end and the domain is growing, how do those cells go to the other end? So that's the question. Very simple question, and we've spent the last 10 years trying to solve it. Now, there is very little proliferation of these cells, and you can very easily show by looking at mean square displacement that diffusion, random diffusion, would take too long because they have to reach the end of the domain inside 24 hours. With random movement, they just couldn't do that. So here was a hypothesis put forward by our biology colleagues. It is known that there's a chemical, a chemoattractant, called VEGF, and that is produced by the tissue. And what happens with the chemoattractant is that cells move up the gradient of the chemoattractant. So the idea was that the cells would come out of the neural tube, they would consume, degrade the VEGF, and that would create a gradient. And then they would move up the gradient, so you now have a biased random walk. And that would lead to the invasion. So that was the model hypothesis. So what we did was we wrote down a very simple mathematical model to describe that. And so I'll just show that here. So C here is the chemical, and this is diffusion. So the domain is only growing in the X direction, and the length here, this is rescaled with the length, and the length here, this is just a function that fits the growth curve. So it's like a logistic growth. This just fits the growth. And this derivation of the reaction diffusion equation on a growing domain is due to Edmund Crampon in his doctorate, co-supervised by Eamon Gaffney and myself in 1999. So when you derive this equation on a growing domain, you end up getting this extra term here, where dot is derivative with respect to t, and this is dilution term. So here we have the production of the VEGF, very simple model here. And then this is the internalization or the degradation of VEGF by the cells. So if a cell is at position xi, yi, then we have this as the degradation term, which is basically a Gaussian that is maximum at the cell center and very rapidly goes to zero as you move away. And so you sum these over all the cells, and this is the total consumption, degradation of the chemical. We need to have boundary conditions. We started by assuming homogeneous Dirichlet boundary conditions for the chemical. Later on, we use zero flux, and our results don't depend on those. The cells we model as discrete disks, and they stochastically sense the gradient and move up the gradient. So what you're going to see here is a video. Oops, if this works. I'm going to get rid of that. Oops, sorry, so I don't know, get this, sorry about this, trying to get this video to work. Oops. So I don't know how to get back my okay, so anyway, sorry, I just does anybody know how I can get my pointer thing back? So that's the laser pointer, but get my get back to my.
So probably if you exit presentation mode.
Okay. All right, here we are now. Okay, this is working. Okay, great. Yeah, sorry about that. So what we're going to do here is here's the domain. Oops. And the domain is going to grow, and cells are going to come in, and the lines you see are the cells sensing the gradient. Oops. Okay, so the domain is growing, the cells are coming in, and you can see this is working really well. They're creating the gradient, and they're moving up the gradient, and it's a directed movement because of the biased random walk, and they're invading. So this is exactly what the model said should happen, but look, something's gone wrong here. These cells have got left behind, very highly localized. So this model doesn't work. So the question about can this chemoattractant work? The answer is no, it doesn't, because cells get left behind. So what we did then was we said, well, if these cells, maybe they stick to the cells in front of them. So maybe there are two cell types, cells at the front that move in response to the chemoattractant, and cells at the back that stick to the cells at the front. And then if we have that model, and I won't show the video, we get this movement happening. The cells do not get left behind. They start to move. So we went back to our biology colleagues and we asked them, are the cells all the same? Because we think the cells at the front are different to the cells at the back. And they said, we don't know, but we will analyze the cells genetically. They analyzed the cells genetically and they found very big differences between the cells at the front and the cells at the back. In particular, they found cadherin 11 was upregulated in the back, and this is the stickiness gene. This is what makes cells stick, exactly how the model predicted would happen. Needless to say, it's more complicated than having just two cell types. So here they did a genetic analysis all along the stream, and they found lots of differences as you move along the stream. So there's a lot of heterogeneity of the cells. So then what we did was a lot of experiments where we did experiments on the computer about what would happen if you moved this type of tissue to here and that type of tissue to there. And they did the experiments in the lab. And mostly we did lots of validation where the mathematical model and the experimental experiments agreed. But more interesting is when the mathematical model and the experiments don't agree. So here's one such example. What would happen if you took followers from a host, from a donor, I should say, followers from a donor, and put them to the front of the host? So you put these new cells here. Our model predicted that these cells would just be advected by the growth because they're not chemotactic. But here is what they found biologically. So here the host in blue, and in pink is the donor. The donor cells became active and they moved up the gradient. So they changed from being followers to being leaders. So this is called phenotypic switching. They switched their phenotype. So the next obvious experiment is what would happen if you put some VEGF along the stream. So here's the mathematical model of what will happen if you put VEGF along the stream. So first of all, here's normal. The cells are invading. And now here's what will happen if you put the VEGF here. The model prediction, which is obvious, these cells turn from being followers to being leaders and move up the gradient of VEGF, and the stream splits. And here is the experiment. They put the VEGF here, this is that red thing, and the stream splits into two, just like the model. So another question they asked us was how many leaders are optimum? And this model cannot answer that question because the parameters, we don't have sufficient data to calibrate the parameters. But what we can do is we can say qualitatively what would happen if you increase the number of leaders. So here's the mathematical model, here's the normal one, and here we've increased the number of leaders. And so it's best to look at this. This here is the path of invasion. And here's the number of cells predicted by the model under normal circumstances. And here it is with more leaders. So we said to experimental colleagues, our prediction is, if you increase the number of leaders, you will get invasion, but fewer cells. And here's the experiment. This is normal. You get the invasion. This is when you increase the number of leaders. You get invasion, but fewer cells. Exactly how the model predicted. But note, the numbers are not the same. Here, less than 10 cells. Here, we're saying 16. But we didn't expect to get quantitative similarity. Now, if we look here, go back now and look at the model, these leaders have gone too fast. And the followers can't stick to them. So then what this model is suggesting is the leaders should slow down and not go too fast. So our biologist colleagues looked at, I don't have I've put in the wrong slide. What the biologists found was that there is something called DAN, which is a gene, DAN, or a protein, I should say, that's expressed right next to the neural tube. And DAN slows the cells down. So in fact, here, DAN is expressed, and so as the cells are moving out, the DAN disappears slowly. So the DAN slows down the leaders so the followers can catch up. And I mentioned to you that these neural crest cells are very similar to tumor cells. So what our colleagues did was showed that DAN could stop tumor cells from invading. So here's an example that by learning about normal development, we can help understand us about medical issues. Now, we're still in the process of exploring how the cells communicate. And I mentioned to you that the cells could be sticking to each other, but this is not the only mechanism by which they communicate. And another possible thing would be, are they secreting some chemical signal? Are they leaving mechanical cues? Or is there tunneling? So in other words, are the leader cells making a gap, a hole in the domain to allow the follower cells to move more easily? And in fact, our biology colleagues found precisely this. There's something called aquaporin, and this is expressed by the cells at the front, and it makes them more polar, and it creates tunnels. So here is an example. Now, if I try and get back now to the ordinary, so I need to maybe come out of this. Okay, so here we have the biology experiment where this is normal. This is where they inhibit this aquaporin, and this is where they put too much of it. And what you see here in normal, they move. In the inhibitor, they move more randomly. And when there's too much applied, they move in a more directed way. And here are the mathematical model. So here you see with this tunneling, you're getting more movement than normal and more movement in normal than the loss of function. So what I should mention about this, that we are still ongoing work here. There are a number of very challenging problems to deal with this work. One challenging problem is that the domain growth, we assumed it was uniform in space, but it's not. It's not uniform in space. And there's a feedback between the domain growth and the neural crest cells. So we're presently looking at that, and we're also looking more at how the cells communicate with each other. But I hope what you've seen there is that with a relatively simple mathematical model, we've been able to test a number of experimental hypotheses, disprove some of them, suggest new hypotheses, and have them validated experimentally and learn more about the biology. So that type of movement is called a leader-follower movement. And angiogenesis, which is the process by which new blood vessels form, is also a leader-follower type behavior. So basically what happens in angiogenesis is that when you get a wound or when there's a cancer and there's a need for, in cancer, the metabolism requires more blood vessels. What happens is a chemoattractant is produced, and it causes cells from the existing blood vessels to leave the blood vessel and move towards the wound or the tumor and set up a new vasculature. That's called angiogenesis. Now, there are many, many models of angiogenesis, hundreds of models. Here's one recently that I've been involved in where it's very, very detailed. You can go take the detailed structure of the vasculature from experimental data and then do, this is work of Miguel Bernabeu and many, many colleagues, including Helen Byrne, Thomas Alarcon, and you can see, you can do then mathematical model of how the blood vessel, how the red blood cells move through. And these are all numerical simulations. So there's very, very detailed models of this, but I'm not going to talk about that. What I'm going to talk about, the simple question that how do we write down a PDE description of this leader-follower behavior? So here's what happens. We have the tumor, which is the source of the growth factor, and in fact, the growth factor is VEGF. Here's the blood vessels, here, and the leader cells are called tip cells. They move up the chemical gradient, and as they move, they are followed by endothelial cells. These cells divide as the leader moves. So this happens through division, and then you get a line. Now, sometimes these cells, these branch, so you get more vasculature, and sometimes they come together. And that's called anastomosis. And now you have a loop, and blood can flow through here. So a model for this is called the snail trail model, and it was first proposed by Leah Edelstein-Keshet in 1982 for fungal growth, and then extended by Baldwin and McIlwain, Helen Byrne and Mark Chaplin, and myself and colleagues. And here is the model. N is the tip cell density. Here is diffusion. Here is movement up the chemical gradient, where C is the chemical gradient. This is the formation of new blood, of new tip cells, this branching. And it's observed that the branching increases as you get close to the source of the chemical. So this has been modeled by this term here. Then this here is where the tip cell disappears because it interacts with the stalk. You could also have two tip cells banging into each other. That's these two terms. Now, crucial to the snail trail model proposed by these colleagues is this is the gradient of a flux. And the flux is what causes the follower cells to divide. So this is the source term. And there's a scaling factor here I will talk about in a few minutes. So this is the snail trail model. So if you take, so this is work by former student Samara Pillay and my colleague Helen Byrne. What we did was we took a cellular automaton model based on these rules, ran the simulations, and then you sum these up and you get this type of density profiles. So here's the tip cells. These are the density profiles of these tip cells, which are these blue things. So you see you've got a high density that propagates along in time. Propagates, propagates, propagates. And then you've got the density of the followers, which are these. And this propagates like a traveling wave. Okay? And the question is this a good description of this? Now, one thing I just need to point out, I don't want to go into the details, this is work of graduate student Duncan Martinson and Helen Byrne. Recently we did to look at the scaling factor here. And I don't want to go into the details of this. It turns out this scaling factor is 1 over the gradient of the chemoattractant. So you've 1 over the gradient of the chemoattractant here. And this was come up, other people have come up with this in a different context, but that's a side issue. So what we're going to do here is we're going to start off with that discrete description of the snail trail and derive a PDE. Okay, now if you think of the snail trail model, what the snail trail model has done, it said individual cells move up a chemical gradient and they move randomly. So we are going to say the cell density moves randomly and moves up a gradient, etc., etc., and that's going to lead to our model. But what we're going to do is we're going to start at the level of the cell and we're going to systematically get up to the level of the PDE. So that's what we're going to do. So we're going to assume very simply that the growth factor is just a simple gradient. This is not realistic biologically, but we're going to take this as the simple thing because what we're really wondering about is more the leaders and the followers, not so much the gradient. So we consider a two-dimensional lattice, square lattice. So if a cell is in lattice site I, j, then if it was just random movement, the probability of it moving in the positive x direction would be a quarter, in the negative x direction a quarter, up a quarter, down a quarter. But now we have this bias due to the chemical. So it's a biased random walk. And this bias we simply take as the gradient of the VEGF, and we take it as a center difference. And in this setup, because it's only in the x direction, gy will be 0, and we just have the bias in the x direction. Now, what about these tip to tip or tip to sprout? These are called anastomoses. We make the assumption that if two tip cells collide, we have this reaction, they become endothelial cells. Tip an endothelial cell, they become endothelial cell. And we assume branching occurs like this, with this probability, depending on the chemical concentration. And so what we do then, we numerically do the snail trail, solve the cellular automaton and get these pictures here, and we must now compare those with our PDE. So the question is, can we get a PDE that will give this type of behavior, which is the averaged behavior of this system, tip and stalk? Okay, so obviously, since this is a random thing, we have to do a number of simulations and then take the ensemble average. And now if we look at the occupancy, and again, this is very standard, if you look at what changes the occupancy of tips in position I, j, well, it could be cells are in the I-1, j, and they move with some probability, move into the region. Or they are at I+1 and they move backwards, or they move up, or they move down, or they move out, etc., etc. And then this is all tip-to-tip anastomosis, etc. This looks very complicated, but it's very, very simple. And standard, I should say, as well. And then this is the equation for the endothelial cells. And then we do the standard idea of going from this master equation to a PDE. By what we do is we write, for instance, this as n at x plus delta x, comma y. And then we expand in Taylor series. Expand all these in Taylor series. This is the temporal change. So this is, we can write this as dE by dt times delta t. And then we do the standard thing of dividing through, taking limits, and this is the model we get. And just to show you here, this is the diffusion equation here, and this diffusion term is this here, h squared, that's what we would normally call delta x squared, and this is delta t. So this is the typical diffusion in 2 dimensions. We say that delta x goes to 0, delta t goes to 0, and if this limit is finite, that's the diffusion coefficient. So what we've done by getting this model is we have related the macroscopic diffusion coefficient, for example, to the microscopic, the probability of movement of an individual cell. So all of these macroscopic parameters are in terms of individual cell parameters. The boundary conditions are zero flux, same as before. And then to get the one-dimensional model, we have to integrate in the y direction. And when we do all that, this is the model we get. And this is the model we've all been using. So let's compare the two. Here you see these kinetic parameters, or kinetic terms, are identical. N squared, N squared, NE, NE, NC, NC. This flux term is the same as this flux term, but there's this term here. Okay? But if we assume, since big N and big E, there we've normalized they lie between 0 and 1, AN and AE lie between 0 and 1. If we're lucky, these might be small, so this might be 1, and then we've recovered the classical model. But look at this equation. This is completely different to the classical model. So this is the classical model, and this is the model that we have derived systematically. It's completely different. And in deriving that model, to get terms that look like these terms, we had to make mean field approximations, and we had to ignore spatial correlations. So now we have three models. We have the cellular automaton model, which is the true solution. We have the classical model, and we have this new model. So here's a comparison. So in black, here's a simulation. In black is the true model, the discrete model. Here's the tip, and here's the stalk. And on top of them are the two PDE models. Perfect agreement. Fantastic. So the two PDE models, completely different to each other, identical behavior. Here's another example. Here's the discrete model in black. And look at the PDE models. Again, a little bit of discrepancy from the discrete model, but very, very close behavior. But once again, the two PDE models agree totally. Now, here's an example where we chose the simulation so that the model wouldn't work. We took initial conditions in the discrete model so that spatial correlations were very important. And the snail trail discrete model gives you this behavior. And the snail trail PDE models give you this behavior. Now, the important thing here is, if this was experimental data, and then you did your snail trail model, you would then say, oh, this cannot be a snail trail because the snail trail model predicts this. But this is the snail trail. But crucially, in this limit, the PDE doesn't hold. And because we derived our model systematically, we can tell that in a way that you couldn't from the classical model. But notice that once again, the two PDE models that are completely different, they totally agree with each other, even though they're both wrong. So to summarize, what we did here was we developed the PDE. What we found was very different looking PDEs give very similar results for these cases that we looked at, but crucially, we're now able to see why the model doesn't work. It doesn't work when the mean field assumption breaks down. And so what we did recently with Duncan and with Kazu Ninomaya, who is a Japanese colleague, and Helen Byrne, we decided, well, why do these two PDE models agree with each other? And what we were able to do was identify two key parameters. And if these two parameters are small, we can do an asymptotic analysis and find that to And if you look at these of tips times branching rate divided by chemotaxis coefficient squared. Now the top line, diffusion of tips, means that they're trying to move away. Branching means they're trying to move in the y direction. The y direction, then the classical snail trail model will not work, and we have to use this more complicated model. And that means that when we look at data, we can immediately see which data can we try to use the classical model, and which data will the classical model definitely not work, and we can try to use this new model. So what we've done in that part of the course, and I'm coming to the end now, or in that part of the lecture, is that we've shown how if you take microscopic behavior, cell behavior, and depending on the assumptions you make on how you go from microscopic to macroscopic, you get different models. And it depends not on the biology or the hypotheses that you've made, it depends on the coarsening approach that you've done. And this is happening in so many areas of mathematical biology. So the question now is, with all these different models that we have, how do we compare them? Are they very different, or are they very similar? Because if they're very similar, we should look at the simplest model. But as you see, they're highly nonlinear and therefore are giving very difficult applied analysis problems. But by systematically deriving the PDE model, we've seen what are the implicit assumptions that are made, and do those implicit assumptions hold? So what I hope I've done, and the last thing is, how do all these models compare with each other? So to summarize, the first part of the talk was really showing how a very simple mathematical model led to new biological insights. And the second part of the talk really designed to show how a biological phenomenon leads to a number of very difficult mathematical questions to answer. So this shows the interplay between biology and mathematics. Mathematics leads to new biology. Biology leads to new mathematics. It's a symbiotic relationship. So I'll stop there and thank you for your attention.
Thank you very much, Philip. I think we should have some questions now. Maybe I can ask the first question. So you have two parameters somewhere in the second part of the talk. You had D and mu, right? Yep. But maybe I misunderstood, but on one of the slides you showed that they were related to each other, D and mu. They were related via the parameter H, no? Is this correct?
Yeah, that's right. So D was mu H squared over 4 tau or something. Yeah, with the PM multiplying it. So the PM is free parameter as well. Yeah.
Yeah. So this H, what does it represent?
So H is the step size.
The step size, right?
Yeah.
So, but then it means that D and mu are they are in some kind of contrast to each other, no? If H is small, is that right? Or there is also dividing by tau. So you are
divided by the tau, yeah. And so this is the classical thing.
They're of the same order in the limit. Okay.
Yeah, because this is the classical thing of deriving the diffusion from a biased random walk that you have to limit. Yeah, and then so the mu and the PM are sort of, they're fixed constants, but they're multiplying the H squared over 4 tau, and it's the limit as H goes to 0. And it's a classical diffusion thing that if H squared,
diffusive scaling, yes.
Yeah, it's diffusive scaling, yeah. So so that's nothing new, we're just invoking that.
Right.
Yeah.
Okay, thank you.
Yeah.
Do we have any questions?
Yes, you have them all in the chat on Zoom.
In
the Zoom.
Okay. Well, the one question is, what, if any, are the barriers you have found when collaborating with biologists?
So the main barrier is the finding a problem that is tractable mathematically, that the biologist can also do experiments on. So this collaboration with the NeuroCrest people, what we tend to do is we sit together and they tell us what experiments they can do in the lab, and we tell them what the model experiments we can do in the model. And then when we decide that there are certain experiments that we can do on the model that they can do in the lab, then basically what happens is they go away and we don't speak to each other. Independently, they do their experiments, we do our experiments, and then we meet together and then show the results. So the big barrier is getting people to do the experiments. Because in many cases, I think experimentalists feel that they can just give whatever data they have to us, and we will automatically give them some insights. What a lot of time they don't realize is that it has to be both of us doing the work, and they doing the experiments. The other thing is experiments are on a much longer time scale. It might take 6 months or a year to do an experiment in the lab that on the computer you can do inside a day. So you do your experiment inside 1 day, and then you've got to wait a year for the results, and that's not very good if you're a graduate student. So those can be barriers.
Okay, thank you. Another question is, with the models that you have shown, what would be the aspects that the cells need to have to be able to detect in order to be able to replicate the behavior observed in experiments and mathematical models?
Okay, so the cells most definitely respond to the chemical gradient, and that is well known. And it's sort of known how they do that. They do that through having receptors that can then internalize and cause things to happen inside the cell that gives them a polarity. But it's still an open question as to what are the other cues. So one of the other cues is something called fibronectin. So fibronectin is a fiber that aligns cells. And it's known that cells will go along fibers. So fibers can give cells directional cues. In fact, that's something we're working on at the minute. It's been shown, and a lot of work on angiogenesis is being done on that, but we're looking at it at the moment from the point of view of the neural crest cells. And so the key thing is then how these cells with each other. And we're still trying to find out exactly what the different types of communications are. The other point is the phenotype switching, going from leader to follower. Now, in the angiogenesis, that's been studied in great detail, and there's something called notch and delta that are two proteins that are within the cell, and it's known that they interact with each other, and high notch, low delta corresponds to one phenotype, and the other way around to another phenotype. In the neural crest, it's still not known how this phenotypic switching occurs.
Okay.
So question.
Thank you. The next question is, how can the mathematical models of the formation of new blood vessels be applied to better understand tumor growth?
So one of the things that we're trying to do here to understand this is that a lot of treatments, tumor treatments, require oxygen. So for example, radiation is more effective when there's a lot of oxygen. Certain chemicals that are applied work best when cells are dividing, and cells will divide if there's a lot of oxygen. So you have this sort of dilemma where if you give the tumor a lot of oxygen, it will grow. So you should reduce the amount of oxygen. But if you reduce the amount of oxygen, your treatments are not effective. So how do you balance the two? That you give, should you give a lot of oxygen so the tumor grows and then you kill all the cells? But what happens if you get that slightly wrong and the tumor just grows? So one thing that mathematical modeling can do, and this is sort of something we're actively involved in trying to understand at the moment, is how does the blood vessels, of course, give the tumor, give the nutrient to the tumor. So understanding how the tumor becomes oxygenated will then help to understand how you can then do treatments, the appropriate treatment. So there's a lot of work going on in that at the moment. Many, many groups are looking at that.
Okay, thank you very much. So there are a couple more questions. Could you comment if the movement of the leader and follower can be relevant for modeling the animal behavior.
Oh yeah, absolutely. In fact, I forgot to put the slide in to say that leader-follower behavior, I talked it in terms of cells, but this happens with animal behavior. You're absolutely right. Yeah, and we can see that also some animal behaviors, you have a switch between leader and follower. And in fact, a lot of mathematical models have been developed to look at swarming, which, you know, and how does a follower sample the velocities around it and then choose its movement? In other words, how does it choose its directional cue to move? That's a huge area of mathematical modeling. And at an abstract level, I'm convinced it's all the same thing. Yeah.
Mm-hmm. Mm-hmm. And what is the importance of the neural crest cells?
So the importance of neural crest cells, well, first of all, if they don't, with these cranial neural crest cells, if they don't invade properly in early development, then that leads to deformities, so developmental deformities. And overall with neural crest cells, there are 66 deformities or diseases, birth diseases, that come from something going wrong with the neural crest. The second thing is, as I showed, the neural crest cells and a number of very aggressive melanoma are identical to each other. And in fact, we saw here that the neural crest cells respond to VEGF, just like the tumor cells, the angiogenesis. Not only in angiogenesis, but also in tumors, you have leader and follower. The aquaporins that I mentioned that were upregulated in the neural crest are also upregulated in tumor cells. So understanding how neural crest invades can help us understand tumor. And when we saw with the neural crest that DAN slowed down the neural crest, we immediately applied DAN to tumor cells, and it slowed down the tumor cells. So understanding the neural crest could really help us understand certain aspects of cancer growth. But we're still a long way off. I mean, that, you know, it's very early stages of that.
The next question is, do cells cluster? And if so, what effect clusters have?
Well, yes, certainly cells do. For example, in early development, a lot of the structures that form are due to aggregations, like, you know, for example, the structures that give rise to our fingers and digits and all these sorts of things. And in many cases, when the cells cluster, this causes their phenotype to change. And therefore they differentiate and become a certain cell type. So the clustering is very, very important in terms of phenotypic changing, but also even if the cells already have the right phenotype, they need to cluster to form a certain structure. And so there's a lot of work in developmental biology with reaction diffusion equations, for example, to understand this pattern formation.
Okay, thank you. The questions keep coming, so there are two more. Can machine learning be useful in understanding cell movement?
So what people are trying to do in terms of machine learning is to see whether ideas from machine learning could help in, say, generating parameter values, you know, because I was talking about qualitatively getting the right behavior, but the next step is to quantitatively get the right behavior. And that will involve then maybe getting lots of data. And then if you've got lots of data, how do you use those data to parameterize your model. And machine learning could help in that. There's also actually where people are using equation learning so that you could say that we think this system arrives from a second order PDE with some reaction terms, but we don't quite know the detail of each one of them. You can learn the equation, and that's what some people are doing as well. So that could be where it could be used to help. But again, this is very early. I'm not involved in any of that work, but this is very early stages. A number of people are looking at this.
Okay, thank you very much. And do your models apply in every type of embryo species? For example, is cell movement different in sea urchins and chicken?
So this is a very good question, and I should have mentioned this. I was talking about the chicken and about cranial neural crest. Even in the chicken, the trunk neural crest that forms the gut, the movement is different. It's driven by proliferation. And in Xenopus, in the frog, the movement is different. It's a run and chase type movement. So thank you for asking that question, because I should have mentioned that the different neural crests in one species seem to have different mechanisms, and between species they have different mechanisms. And this is one thing where I think mathematical modeling might be able to help, because I'm convinced that there should be an overarching framework, like a unified model, if you like, of this movement. And then what we could see is that the different species and the different neural crests are examples of subsets of these models. So I think that having an overarching leader-follower type of mathematical model could give us insights into all these different things.
Mm-hmm. Mm-hmm.
But that's a very good, that's
okay.
Thing.
Yeah, thank you. And I think it will be the last question. How can these models, how can they contribute to the treatment of cancer?
So like I said, that if we could understand the delivery of oxygen, we might then be able to understand how to give alternating treatments. So you might end up saying, for example, this has already happened, that if you give treatment that increases the vasculature and then you give chemotherapy, it works really well. So increased vasculature means more oxygen, which means that your chemotherapy is more effective. So we can use mathematical models to try out all sorts of different combination therapies or sequential therapies, and therefore suggest to biologists which one of these might work. Because as you can imagine, if you decide, if you've got a number of therapies and you do them in a sequential way or a combinatorial way, there are many, many different combinations. And what mathematical modeling can do is to narrow down which might be the more effective. So the experimental people don't have to do millions of experiments. They can maybe do a smaller number of experiments. That's the aim, that's the goal, the dream.
Okay, thank you very much. I think the number of questions illustrate how stimulating the talk was.
Well, thank you, and thank you for the questions and for the interest. Thank you.
Thank you again. And with this, and before passing over to Professor Meade again, let me just invite everyone to the next talk. It's next Tuesday, 25th of May, the same time. And the talk will be by Professor William Phillips from the University of Maryland, who will tell us about rewriting the metric system with quantum physics. Thank you again, Philip. And I would like to pass over to Professor Meade now, who will say a few concluding words.
Okay, thank you.
Muchas gracias. Al revés, yo soy el agradecido con Kirill por esta conducción y con Philip también por esta presentación tan formidable. Para nosotros, un evento como estos nos genera otra vez la convicción de que fue una gran idea generar este consorcio que nos permite, primero, pues compartir los avances con investigaciones en curso. Segundo, que como en este caso Philip o Kirill lo ha hecho, que comparten su conocimiento, que compartan sus investigaciones con muchos interesados de toda la comunidad científica, pero en particular de quienes desde el consorcio se pueden sumar a este análisis, a este estudio, a esta investigación. Es además muy motivante porque pues una presentación como la que hoy nos hace Philip acredita que las investigaciones, siendo muy profundas en un solo campo, pues ofrecen horizontes de investigación en un amplio espectro de especialidades. Pueden participar médicos que ojalá hayan visto esta plática, ingenieros, químicos, incluso genetistas. Todas estas especialidades podían ser motivo de análisis y de estudio. Y uno de los propósitos que nosotros hemos derivado o hemos querido tener de estas presentaciones es que haya muchos jóvenes que se interesen por la ciencia. Y yo creo que todo lo que hoy nos ha dicho Philip es particularmente desafiante, es particularmente inquietante respecto de lo que el conocimiento nos permite saber. Yo creo que una de las acciones de la pandemia y uno de los imperativos de la nueva realidad es que hay que respetar la ciencia. Y para respetar a la ciencia hay que conocerla, hay que investigarla, hay que trabajar para ir desentrañando sus secretos. De manera que esto, pues, para nosotros resulta muy estimulante. Ojalá muchos jóvenes se interesen por participar con sus estudios y con sus opciones profesionales alrededor de estos estudios y subrayar una vez más cómo la universidad, y ahora sumada a todo este consorcio de universidades, van siempre a la vanguardia del estudio, a la vanguardia del conocimiento, a la vanguardia de la investigación científica. Sumados el esfuerzo de todos, podremos ir construyendo un mundo mejor, jóvenes mejor preparados y una realidad que ofrezca un mejor horizonte en el futuro para toda la humanidad. Gracias, Kirill, gracias, Philip, y gracias a Jaime por su conducción, por su talento y por su participación tan activa en estos eventos que estamos celebrando martes tras martes. Felicidades de nuevo y le damos la palabra a Jaime para cerrar este evento. Si no pudiera, le pediríamos a Araxi que nos ayudara a hacer este cierre.
No, pues nada más, muchas gracias a todos por acompañarnos y los esperamos la siguiente semana.
Los alcances de la ciencia son cada vez más audaces e innovadores. Las nanociencias nos conducen al microcosmos, un mundo imperceptible para el ser humano, en donde se estudian, se diseñan y se aplican nuevos materiales y diminutas herramientas para usos prometedores. El avance de la nanotecnología brinda posibilidades de mejorar la medicina y la salud, así como de aportar opciones para el rescate y conservación del medio ambiente.
En Fundación UNAM seguimos trabajando por la cultura, la ciencia y la educación. Sin salir de casa, reflexionarás con los martes de ciencia a través de las conferencias Universidades por la Ciencia, que emitimos en conjunto con el Colegio Nacional. Y para disfrutar, regresamos con la segunda edición de los ciclos de documentales de Jueves de Ciencia, con la transmisión especial de TV UNAM. Además, conversamos sobre nuestra nueva realidad y sus afectaciones en distintas disciplinas a través del Corredor Cultural de Autonomía. Sigue nuestras redes sociales y no te pierdas ningún detalle en www.fundacionunam.org.mx.